is Lucie. I'm a master's student here at the University of Hawaii Manoa, and my supervisor for Sea Grant is Eric DiCarlo. And my topic is heavy metals in the Ala Wai watershed, and I'm going to focus specifically on copper today. And actually, I will be defending my master's thesis on this topic in less than 2 weeks, so thanks for letting me practice a little bit, and wish me luck. Right. So to start off, I want to say something about what heavy metals actually are. The definition is kind of wishy-washy, but they are basically just dense metallic elements. And some of the examples we might know from everyday life are lead, cadmium, iron, copper, which I'm going to talk about today, and zinc. All heavy metals are toxic to most organisms when they are ingested in large amounts. But some of them are also important nutrients in smaller doses, basically. It really depends on the metal. Iron, for example, is a really important nutrient that humans need to make red blood cells, whereas lead is dangerous to us in basically even the smallest amounts. It's never a good thing. I'm focusing on copper in my thesis, as I have said. Can any of you maybe guess which of those two categories copper is in? Is it also good, or is it only a bad heavy metal?
They split.
Yeah, that's right. It's a micronutrient, so in small doses it's important to most organisms, but it gets very toxic very quickly when the concentrations are too high. So I'm focusing on heavy metals and streams, and when we look at water bodies, there are two main questions that come up in relation to heavy metals. The first is, where do they come from? And the second one is, how are they being transported by the river or stream? So are they dissolved in the water, or are they in particles? And those questions are important to answer to figure out if those metals are natural or human-made, and also to figure out if they're potentially dangerous or not. So many of us would probably think that copper, say in the Ala Wai canal, which we all know is really pretty gross and polluted. So you might think that almost all the copper in there is just from human-derived activities, but there actually are many natural sources for heavy metals, such as copper, as well. So first of all, as the stream forms and flows through the mountains here, it kind of dissolves the rocks in a process called rock weathering. And a lot of the material that's dissolved in streams is actually derived directly from this process. And a lot of the particles in the streams are just basically little rock fragments being washed away. Then the next important source is soils and plants. Soils are made up of the altered rocks basically after they've been weathered. And their whole chemistry is a lot different from that of rocks because it's more dominated by living microorganisms and plants that kind of dominate this chemistry and take up and release different chemicals, such as copper as well. Then agriculture influences soils in a really major way. There are a lot of fertilizers and pesticides who contain heavy metals and can run off into soils and also indirectly into streams. Then when we enter areas where humans have built entire roads and cities on, the natural sources become really small and kind of get overwhelmed by all of these human sources, such as traffic and construction. And finally, many industrial processes also release heavy metals into the environment, where either they end up directly in streams or indirectly through soils or through the atmosphere when they get released into the air and then deposited into water bodies. So we can see there's really a lot of different sources that these heavy metals can come from. Now, the reason I decided to study copper in the Ala Wai watershed is that it used to be a concern mainly in the early 2000s. There were multiple studies that discovered that, for one, in the Ala Wai harbor, copper was actually exceeding the Hawaii state acceptable water quality level. And also fish in Manoa Stream, upstream from the Ala Wai, were found to have high copper burdens. And copper was high in soils and road-deposited sediments all throughout the watershed as well. So since many of the human-made sources of copper that were in use then are still in use today, such as these three pictures that you can see, metal plating, boat paints and brake pads from vehicles. These are all still in use today, so I was wondering if this pollution was still a problem. And my study area, this is a map of my study area, has a really nice transition that includes most of the different natural and human-made soils I was just talking about. I started my San Manoa Valley in the little mountain streams that are pretty much untouched by human activity, and went through the urban areas of Manoa, through Honolulu, and into the Ala Wai Canal to kind of capture all of these different potential sources. These 3 photographs are all from stations that I included in my sampling. They're a little pixelated, I'm sorry. So they illustrate that there's a big change from the pristine mountain streams on the left, which is way in the back of Manoa, to the urban channels. That's the middle picture. That's right by the UH campus, down into the Ala Wai Canal. So from the beginning of my study, I was expecting that heavy metals would change between untouched mountain streams and the urban Ala Wai Canal. And I was also expecting that there would be natural sources in the back of the valley and more pollution in urban Honolulu. Then another interesting thing about the water cycle here in Hawaii is how important the rainstorms are. They're really, really important. They transfer a lot more material from land into the coastal ocean than the base flow of the streams. And that's although they are so short in duration. If you look at it over a year, actually they dominate the material transfer. So they wash out more to the ocean, although they're only such short events. So they're really important to look at if you want to look at pollution. Yeah, in the back of Manoa, for example, the flow of the river, so basically the amount of water going, can increase 60 times in only 15 minutes, which is why we all get these flash flood warnings on our phones. Like 2 days ago, which is when I took these videos on the Manoa campus, actually. So to find out how copper is different between those low flow conditions and the storms, and also between up in the watershed and further downstream, I collected 3 different sets of samples. The first one is from stations along Manoa Stream, going from those pristine stations in the back down through urban Manoa, pretty much to campus. The second one, so it's going from natural to urban. And the second one is in the channel that's leading up to the Ala Wai, as well as the Ala Wai Canal itself, which I'm expecting to have more of a pollution and signature and also some seawater influence, actually. And the third set is old samples. They're about 20 years old, from storms in the late 1990s and early 2000s that were collected by my advisor back then. So I'm going to compare those as well. And these are my results for the copper concentrations. The 2 top bars are the back of Manoa Valley, the kind of pristine stations. And the 2 bottom ones are the channel that's leading into the Ala Wai. And the orange and the blue are both bars for low flow conditions, and the green are the storm conditions. So there are 2 important things that you can see from this graph. The first one is that the copper increases from upstream to downstream stations, no matter which flow conditions. So both for the low flow and for the storms, it gets higher downstream. And the second one is that for each station, there's also an increase in copper from low flow to storm conditions. So basically what that means is that the copper always gets higher as it flows down through Honolulu, and it also always gets higher everywhere when there's a storm. So then I asked myself, what's the difference between the stream flow from when there's a storm compared to when there's not a storm? This is a schematic of a stream and the different sources of water that it's made up of. Because the stream is not only made up from rain that just falls into the streambed and then runs off, it actually also has a big component that goes through the soil and the rock and becomes groundwater before coming back into the riverbed. Because otherwise the stream would always be dry when it's not raining, which is not the case. And it turns out that during low flow, the groundwater is actually the most important part of the stream flow. And most elements that the stream carries during these conditions that are dissolved in the water come directly from this rock weathering and dissolution that I was talking about. But during storms, this groundwater is overwhelmed by the surface runoff, which becomes so much larger because of all the rain that's coming down. And that water is just full of all sorts of things, like dust and dirt from the roads and the air, soil water from the surface, and plant debris. So although during storms this is the same stream, it's basically a completely different kind of water that we're seeing. And now we know from the data that I just showed you that the surface runoff during storms has more copper than the groundwater runoff that occurs during the low flow conditions. But that doesn't really tell us where the copper is actually coming from. To figure that out, I am using a tool called Isotopic Measurements. And I put this little detective there because it really oftentimes feels like you're looking for the fingerprint of some sort of criminal when you're looking at this kind of data. The whole technique is based on the fact that for many elements, there are different variations of the same element called isotopes that are only different because they weigh a little bit less or a little bit more. And this is the case for copper as well. We have copper 65, which is a little bit heavier, and copper 63, which is a little bit lighter. And we do have instruments that are good enough to measure the difference between those two kinds of copper, but it's really a very, very small difference, so it's very hard to measure. And I had to spend many, many hours in the lab to be able to do it. So if that's such a hassle, then why should we do it, is the next question. And the reason is that those isotopic measurements can really help us kind of find the culprit in this detective scenario that I just mentioned. As an example, plants, they do take copper up as a nutrient, like we said before. And when they do so, they actually prefer to take up the lighter copper. And so if we then find a lot of light copper in the stream, for example, that I'm looking at, that could indicate that this copper is coming from plants. So that way, copper isotopes can tell us about the sources of copper when we look at if there's more light copper or more heavy copper in the sample that we have. And in my case, I found an interesting difference in the copper at Waiakiakua, which is my most pristine mountain stream kind of station. It's the furthest back into the valley that I sampled anything. And here during low flow, there was only very little copper, as we would expect. It's only the natural copper sources from the rock dissolution. So very little copper, but it was more of the heavy isotope. Whereas during storm flow, there was a lot more copper, and this was more of the light isotope. So there's really a difference, which tells us that during storm runoff, there's an additional copper source that has more of the light isotopes. And that could be from soils, agriculture, and plants. So from these more shallow kind of sources in the ground. So that's one of the possibilities of using isotopes for these kinds of studies. And now this last plot is a little bit hard to read, but bear with me because it shows us where we stand in comparison to the rest of the world with copper pollution. So, So the top box are my samples from Oahu. It's basically the same plot that I showed you before, where it goes from the low flow in the back of the valley to storm flow in the back of the valley, and then low flow downstream by the Ala Wai, and then storm flow in the Ala Wai. And then I have comparisons from all of the different continents, and we can see immediately that there's a really wide range that my samples have, right? So the low flow, the little orange bar in Manoa, is way below the world average, which is the big blue bar on the top. But the storm flow that enters the Ala Wai, the long green bar, is way above that and actually comes closer to the Seine River in France, which is a really polluted, very big river. So we can see that just a few miles down the stream, as well as a few hours of difference between if it's raining or not, can really make a gigantic difference in the copper pollution that we see in the Ala Wai watershed. And that's pretty much it. Down there is me doing lab work some of the time, and then most of the time on the right, luckily. And that's my supervisor, Olivier Rouzet, and I want to thank him and Eric DiCarlo and Chris Measures, who are also on my thesis committee, and then Sea Grant Hawaii, of course, for paying my graduate assistantship for 2 years and teaching me all sorts of great stuff. And IFREMET, which is an institute in France where I do measurements. Thank you.
Hi everyone, thank you for coming out today. My name is Evan Barba. I'm a graduate student at the University of Hawaii Manoa. I both live and work at Moku'oloi in Kaneohe Bay at the Hawaii Institute of Marine Biology, so out on the windward side. And today I want to talk to you a bit about a project that I worked on over the summer, bring you, south from Hawaii down to Jarvis Island in the equatorial Pacific. And I want to talk to you about some of the small critters that you see out on the reef that typically they're kind of hiding in rocks, but they're important little critters. Hopefully you guys get some appreciation out of them. So where is Jarvis and why is it important for any kind of studies related to changes in temperature? So Jarvis Island is located at 0.2 degrees south, right on the equator. If you draw a line south from Hawaii, you might hit it. It's a very small island. It's actually only 4.5 square kilometers, so barely bigger than the size of Waikiki. Small little isolated island, but what makes it great for studying anything related to changes in temperature in the seawater was that it experienced a really, really hot summer in 2015. So these dark colors on the plot indicate a water mass that was really, really warm for an extended period of time. And as you can see, Jarvis falls right in the center of it. And to give some context into how warm it was, this plot on the right shows temperature at different months. And each different color on these series are years that had abnormally warm temperatures. And this bold red line represents the 2015-2016 summer. They had very, very warm water that remained there for a long time. And so what does warm water mean to a reef? Well, sometimes it can be shocking. So let's look at some visuals. April 2015, scientists went down to Jarvis. They were working on environmental monitoring, and this is what the reef looked like then. Just beautiful corals as far as you could see, plating corals down the reef. The entire photo covered in coral. But in May 2015, that water started to come through, become a bit concerned about that warm water mass staying out on the reef. So they returned in November of the same year to try to document what was happening on that reef. In 2015, different. This white color doesn't necessarily mean the coral has died. It's just undergoing a process known as coral bleaching. This happens in water for a long period of time. So corals, they're an interesting animal. They have a really close relationship with a small little plant that lives inside of them, a little algae. And so that little algae almost acts as solar panels for the coral. So it's a nice little trade-off. The solar panels, the coral or the algae, they get free rent living inside of the coral tissue, and they provide extra sugars that the corals can use as energy. So it's a nice little trade-off, pretty good win-win. But during warm periods of time, that relationship starts to break down. And the coral symbionts don't live inside the tissue after that. So then you end up with this white color where the coral tissue is actually clear. So what you're seeing is just the exoskeleton that's there. And where corals die in this situation, so there's hope for recovery. But in this instance, scientists returned in May of 2016 to see how this looked a year after the event had begun. And what had been feared was kind of played out. The reef was totally changed. So when they're exposed to these warm temperatures for too long that they can withstand, a lot of times, if it gets that severe, the coral can die. And this is something that you can compare to deforestation or just clearing out trees. So in that situation, the tree kind of acts as a for the rest of the ecosystem. When you get rid of the trees, anything else that depends on it for a home ends up in a situation where they don't have their habitat. So in this coral reef instance, when the coral gets taken out and dies, it breaks up. You end up with this reef-scape-wide change, like landscape change. So I compared to deforestation at this scale. And what they saw in 2016 was around 95% of the coral on some of these small reefs had disappeared. And you're left with a lot of algae, just thick mats of algae in a very different reef. And this can have big well-studied fish and obviously the coral. But what we were curious about in this project was really looking inside the reef at everything that's hiding that you typically don't see when you're out snorkeling. There's so much life inside the reef that gets more or less ignored because it's very difficult to study. So here's an example of some of those critters that we saw. I just want to take a moment to appreciate the vast, different amount of color and function, beautiful creatures that you see if you really look close into the reef. So if you're just out snorkeling and about, most of the time you'll see everything on these walls. You get beautiful pictures, but all the tiny stuff living inside the reef, surprisingly, that actually makes up a majority of the number of species on a reef. In Hawaii, they've estimated that to be upwards of 85% of the number of species on a reef are actually these really small animals, these little critters living inside and among the reef. And just for scale, some of these are the size of a grain of rice. The shrimp, about the size of a grain of rice. It's small, but beautiful. And we don't know very much about them. So how do we start to learn more about them? Well, I was part of a team on a research cruise that visited Jarvis among some other islands last year. And what we were doing was using these settlement structures known as ARMS. They're autonomous reef monitoring structures, clever little acronym that someone came up with. But they have these all over the Pacific. They're little PVC units that you can take apart. So you put them out on the reef, you can take them apart and study them later. And so the general process of these is you'll go out to a reef, you'll put out one of the units, and on the left you can see a fresh unit, nothing settled out on it. And we'll put them out with big, heavy weights and a lot of zip ties because these remain down under the sea for up to 3 years or more. So these will withstand 3 years of ocean conditions, whatever storm comes through. We want these to be able to be recovered later. And so after 3 years of being left out, you have this essentially an artificial reef structure that mimics reef habitat that things can colonize and live on, and all the critters can live inside. And then so to go down and get them, I was part of the dive team that would go down, take the weights off, and then we'd put one of these crates that you see here that's lined with a really fine mesh so nothing can get out. You put them on top, strap it up, and then take them up to the surface to be able to contain everything that's inside of this unit so we can see all of the little critters without taking out part of the reef. So once we have them up and ready into a larger area, we can look at them for some field processing and get as much done while we're at work before bringing any of these organisms back. So we can do this while they're live. And so I was out on the NOAA ship Ala Wai, which has a lot of great lab space for us to be able to process as many of these critters as we can while we're out. So there's 3 main categories that we would separate this work into. So once we disassemble the unit, we can take the plates, the individual parts of that stack, and look at what's attached to it. We can also get all of the little critters that are less than 2 millimeters, tiny things that would be really hard to identify by eye, and sort those from the larger critters that would be greater than 2 millimeters. And each of these different steps, we do some different methods to figure out what's inside them. So specifically for the plates, we'll get photographs in really high resolution that we can come back to later to determine the percent cover of what sponges or different types of algae, oysters, all the little organisms that are attached onto these plates. But then if we want to get more detailed identifications of them, we can use genetics. And it's really a pretty powerful tool. So the way this process works is we'll scrape everything off, put it into a blender, extract the DNA. So we extract the genetic material, and then we'll bring it through computer programs after it's sequenced to be able to get a list of what's inside that sample. And one way to think about this is like you're at the grocery store, fill your cart up with a bunch of food, it goes through and each item gets scanned, right? They all have a little barcode, and that barcode is unique to the product that it's matched to, a loaf of bread, box of cereal. And then at the end, you get a printout with a receipt, right? Has everything listed on it. Essentially, we're getting our best identification on a receipt list at the end of what's inside that sample. So it's kind of a nice way to use genetics to study a lot of things that are hard to identify. So after we have that information, then we can look at some of the mobile critters, everything that's crawling around inside that's not living attached onto the units. So we'll use the same process for all the tiny things that we can't identify by eye. Once it's smaller than 2 millimeters, it takes a lot of effort and some pretty impressive microscopes to actually be able to determine what those are. So we'll use genetics to aid us in the same way that we do with the plate samples. But then the other process that we do in the field that ends up being probably my favorite portion of this type of work is looking at the things that are the large critters, greater than 2 millimeters. So a lot of them are still pretty small, but the way we'll do this is put everything into a big tray, sort it all out, put like with like, and then actually count and photograph all of these little critters. I think over the course of 2 months, I took 15,000 photos. And the reason we take these photos is we want the best captured picture of when the critters are alive. So when they're actually alive is when they have the most color, they have the best expression of them, as opposed to being in a jar of preservative, right? It's hard to really tell what something is once it's dead in a jar. So we get these really high resolution pictures that we can send to experts and get the best identification possible. So we'll narrow it down the best we can in the field. But when you're working all the time around the clock, it's really hard to be an expert in every single type of little critter, especially when there's so many of them. So we do our best in the field and then send them out to someone who's spent their whole life studying crabs or their whole life studying worms. And that really aids in getting solid identification that we can then compare to genetics and as we did in the others. So just to highlight a few of my favorites here. Can anyone tell me what any of these critters are? Yeah. Which one? Sorry. Yeah, so that one's actually a type of flatworm. It's very closely related to the Spanish dancers. Spanish dancers are a sea slug that's really close, but this, I can tell how you can see that because it's actually kind of fun. These two little appendages here look like the Spanish dancers' little rabbit ears. And it's actually a flatworm that mimics those Spanish dancers. You got one? Yes, that's a crab. Good job. Yeah, it's called a porcelain crab. And that was probably one of my favorite critters I saw this whole trip. But this one? It's a flatworm. So closely related. And if you look closely, its eggs are actually bundled right there. It's pretty cool. Yeah, and this is probably right on that 2 millimeter mark. Really small. Tiny little thing, but you can, with these photos, you can end up getting good identifications. So back to Jarvis. Once we know who's there, that's when we can start having some informative discussions about what's going on in these communities. And so these charts represent what we've seen in Jarvis in the past, with this left column representing Jarvis from 2015. And each bar is a percentage of the total number of organisms that are out separated by group. So like crabs, snails, different taxonomic groupings is what it's called. And one thing you can ask is, do those proportions shift? Do you have more crabs, less snails? What's shifting in terms of group? And we're still in the process of identifying who are in our samples. So we haven't got to the point of being able to analyze that yet. But another way that we can think about it is, what's their role? Do they eat algae? Do they eat coral? And are the proportions of those groups changing based on what's available to them now that there's more algae in the reef? Are there more organisms that eat algae? Or what's driving those types of differences, so we can see if the warm water impact and the amount of coral actually impacts these little communities that live within the reef. And the real importance of work like this is this was a good opportunity to look at a window into the future in terms of what our reefs might be facing. This was a very short event, a year, and the reef went from almost 100% coral cover and was reduced by 95% in some of these reefs. So a lot of the coral has gone a very rapid change, and we're currently experiencing rapid declines in coral reefs around the world. Whether or not it happens in a span of a year is definitely a different question, but this gives us the opportunity to see a very rapid shift between these two time periods. And it gives us a chance to try to understand more about some of these organisms that we really don't know much about. We know they're there. We know they provide a lot of ecosystem services. They're eating algae. They're recycling nutrients. We know they do a lot in the reef, but we don't really know who they are. So that's a good step in determining why they may be important or whether they're resilient to changes in the future. And with that, I'd like to thank you for your time and thank you for listening to me share some of my research. And of course, acknowledge all of the funding and field lab and data analysis help that was involved in this project. And I think we can open for questions fairly soon too.
Okay, so we have 10 minutes. We have 15, but we actually have only 10 minutes for questions because this is part of their training. We need to, Maya and I will evaluate them once they're done with this training, with this Q&A session. So we do need a little time with them. Barb?
Yes.
Yeah.
The one that went to Jarvis.
Yes.
Okay, so the reef that you showed us from April 2015.
Yep.
Were the little animals living in the reef, you've now discovered, were they ever studied then?
Oh, yes. So we can compare to some units that were put out in 2015 to ones that was just before the bleaching event. Sorry, I glossed over that again. Excited about the little critters.
Yeah.
But yes, we put units out in April of 2015, and then they were collected this summer of 2018. So they were out for 3 years, and the ones prior were put out in 2012. So we have a snapshot of pre-bleaching in 2015, and then post-bleaching where there's a lot less coral cover, and the units that were just collected, they experienced a totally different reef. And so it'll be interesting to see how those communities changed. I don't have any full statistical, anything fully analyzed, but anecdotally, I can tell you that they were different reefs. And we got a much lower count. Yeah.
And then for the young lady, you had mentioned the copper increases that are in the streams. What about as it enters the ocean? What effect does that have on our ocean?
That's a really good question. Actually, I didn't sample all the way into the coastal ocean, but since the ocean is that big compared to the stream, it is diluted tremendously. So the copper concentrations, even if they're a little bit higher just where the Ala Wai enters the coastal waters, it doesn't change much beyond the mouth of the Ala Wai, really. And there also are processes going on in where saltwater and freshwater mix in an estuary that usually lead to heavy metals kind of attaching to particles and going into the sediments. So I would say most of that pollution ends up in the sediments of the Ala Wai Canal as opposed to the actual coastal ocean.
Is there a baseline for how much copper is in our lava?
I can't tell you off the top of my head, but there is quite a lot actually compared to other rocks for sure. But what we see in the little orange bar, if you remember, that's pretty much what comes from that.
Okay.
Yeah, I had a similar question. Background property. Why being volcanic versus continental?
Yeah, it's totally right. You're right that there's a lot of copper, and some other trace metals are actually even more abundant in basalt, like chromium, for example, is very, very high here. Vanadium, a bunch of things. But as I said, since you can see during the low flow, what's in that stream, that's basically what's coming from the basalt. But anything above that is really mostly from anthropogenic sources or surface
So
runoff.
continental versus volcanic?
That makes, yeah, it makes a difference for
Yes,
sure.
more.
The volcanic islands have more trace metals on average, I would say.
Did you have, are there other locations or sites throughout the Hawaiian Islands where copper has been measured in similar, more pristine environments?
Not that I know of. I did also analyze samples from Tahiti, which is relatively similar and a little bit more pristine, and they were even lower in copper than the.
What is the ideal temperature for coral?
It depends on location and species. So it can be highly variable. Certain corals can thrive really well, upwards of 32 degrees Celsius. Some of them will make it. Some of them will die at 29 degrees Celsius. So it can really be variable depending on where you're at in the world. Certain populations, like Hawaii, for instance, we're up in colder waters compared to a lot of the tropics since we're pretty high latitude. So a lot of our corals might be more sensitive to temperatures that might be experienced at the equator often, where those populations might be a bit more adapted to warm waters. So it really depends on where you're at and what species you're interested in.
By the Fahrenheit temperature?
Yes. A quick mental math, like 85, 86 is where it starts to be a bit warm. Yeah.
In your Jarvis Island temperature, are you measuring surface temperature? Or subs?
They actually have sensors out that monitor at different depths across the island in different locations. So they'll have 15 feet, 30 feet, and some of them all the way down at about 100 feet. And so they have a pretty good gradient in the north, east, south, and west portions of the island. So each of the sites is located at, has different depths with temperature.
How about the rest of the ocean?
The rest of the ocean, it depends
I
on,
mean, you had a map of.
Oh, a lot of that was actually derived from satellite models. And so people can estimate temperature based on surface temperature. Yeah. So the map provided was surface temperature, but we do have ground truth sensor data for the sites that we were interested in.
Some of the corals we've had bleaching here on the island.
Yes.
I've heard a speaker from Coconut Island talk about some of the corals seem to be mutating to handle the warmer temperatures because they're finding, a bleaching effect, but yet there are surviving corals within it. So you're doing studies on them.
Yes, there's hope for adaptation. Yeah.
Is there a possibility in Jarvis that there will be corals that will come back, or because it was completely obliterated? Or, you know what I'm saying?
It was very heavily reduced, but that's something I can't fully speak to. There's a lot of debate around whether and how Jarvis may recover. There are other populations that may be supporting new recruitment of corals coming back to the reef versus ones that are currently there being able to seed the rest of the reef. So there's a lot of debate about what will happen. It's still a process everyone's trying to understand in time, but with these events happening more and more, it becomes a challenge to the coral. And this is a good example of how it can happen quickly. Once it hits that threshold and goes over their limit, they can really be impacted. So there is a chance for a very, very strong chance for corals to be able to adapt to warm temperature. But it depends on how fast the temperature is changing.
Has the corals on Jarvis, have they recovered since 2000?
I was just there in 2018, and unfortunately, there are fewer corals there than there used to be. It's a big loss to the Central Pacific coral reef community. That area, it really experienced some warm water that took an impact, and it's really unfortunate. But we will see what the future holds. Yeah, and it's a good kind of canary in the coal mine. It will happen in places if the ocean continues to warm. So it's something to be aware of.
Back on that copper topic, so rather concerning, right? The amount of copper and storm runoff. Is that, do you guys have any ideas on why that is? Is it just attributable because it's a smaller stream and therefore you have just a lot of that concerning versus a lot of those larger rivers? Or is there any, you guys have any thoughts on why that's such a high spike?
I would say if you look at, so the river that it was comparable to was the Seine, which is like a really large river in France, right, going through Paris and stuff. I think the big difference is that the streams are very small, like compared to these rivers, and they're much more susceptible to sudden changes in their chemistry because of the small amount of water and the big difference between the low flow and the storm flow. While a more established river and like the Seine would probably doesn't experience these quick changes at all. So it's more like a sudden flush of all of the trace elements such as copper into the storm runoff when these storms happen, which is a pretty not unique but a special situation here for sure.
Was that sample for the comparison for the Seine from the middle of Paris? Or, I mean, it's quite a long river, so.
Yeah, it was in Paris. Yeah.
Gentlemen, in Hawaii, what's our normal water temperature for the ocean and how much higher has it gone because of global warming?
Well, that's a tricky question because it does vary in kind of large scale as well. So the big events that would be more concerning are like during El Nino, when you have warmer temperatures driven by different processes as opposed to just the long-term increases. So that's really where it gets a little more dangerous. That's where we saw bleaching in 2015, 16, and minorly in 17. And in those periods, it was several degrees warmer for an extended period of time, but not enough to quite cause massive bleaching and death. So it hasn't gotten to that point, and we're currently not experiencing a severe El Nino. So we will see, but it has increased in the past few decades, at least for sure.